Good morning, and thank you for coming. I should say thank you all for coming, since we have a guest who hails from the great state of Texas. Yeah, so it's my pleasure to introduce Dr. James DeGregori, who is a longtime friend and really a hero. He has been a pioneer in really helping us to understand cancer from a perspective that goes beyond mutations and really into the environment that's required for cancer to grow. So James did his, well, like me, he served time in Texas. He did his undergraduate at what some people call that other university in Austin, and then moved on to PhD at MIT and a postdoc at Duke before going to the University of Colorado, where he has risen through the ranks and now holds the Courtenay, is that correct? Courtenay and Lucy Patton Davis Endowed Chair in Lung Cancer Research, and is a deputy director of the University of Colorado Cancer Center. And among other things, he is the editor-in-chief of the journal Aging and Cancer. So I'm very pleased to have James have joined us today.
Great. Thanks so much, Jaime. It's great to be here. I've never been to Minnesota, so this will be a first. And I'm going to have a stopping point in the middle for questions, but feel free to ask questions along the way as well. So I know the title sounds a little bit depressing, but if you work on aging and you don't have a Black sense of humor, you're going to, get very depressed very quickly. Let me see where there it is. Okay. So I'm going to start with something that hopefully everyone already knows, that about 90% of cancers develop after the age of 50. And so here we're going to really ask the question, well, why is that? And, from the conventional point of view and from even what's still in textbooks, this is just the time-dependent accumulation of mutations that accounts for this. And I'm going to try to convince you that that's overly simplistic to the point of being quite wrong. And I think it can lead to a lot of misunderstanding and of cancer evolution. And it's not really an evolutionary way of thinking of disease. And so one of the ways we address this is we were trying to understand this sort of quandary of why it is that, different cancers that have an aging dependence can be driven by very different number of driver mutations. So for different leukemias, they vary by several fold. And for solid cancers, they vary by as much as 10-fold in terms of the number of characterized drivers. And of course, this isn't really taking into account epigenetic changes and chromosomal rearrangements and other type of drivers, but suffice it to say that different cancers in our bodies are driven by very different numbers of drivers. And we also know that cancers arise in stem cell pools that are of very different numbers and very different organizations. And just showing two examples here, gut stem cells, you have roughly 10 to 20 of those within any different individual crypt. And you have, up to 10 million of these crypts in your body. So 100+ million gut stem cells in your body. And hematopoietic stem cells, there's roughly 200,000 of them, and they're relatively intermixing. And yet, despite all these differences, if Andre Rorschach did a very simple thing, he just basically normalized all these to their maximum incidence, put that at 1. And you can see these different curves fall right on top of each other. And I think this is telling us something very fundamental that I'll be talking about today, that has to go beyond simple mutation accumulation. And so in this Eli paper, he developed a mathematical model to test the conventional sort of mutation-dependent accumulation of mutations as the cause of this age dependence of cancer, versus a model where you had differential selection depending on the point of life. And what he found is that you really needed this differential selection. And the way he could reconcile all these differences was by having selection be negative in youth, but become positive later in life for the very same mutations. And this just shows you different parameters that he had used. And it turns out that most of them worked pretty well. It didn't really matter so much how negative or how positive it was. But I'll get back to this later on, because it turned out to be a fortuitous exercise, because later we could really put mutations on this curve and show that different mutations have different behaviors in this respect. So then the question is, why do we get cancer and other diseases mostly at old ages? And indeed, why do we age at all? And I think we have to step back and think of the evolutionary reasons. And hopefully most of you are already thinking this way, but I'm amazed when I teach students, this is the last thing they'll come up with when you ask somebody, why do you get cancer in old age? And it's simply that there's minimal selection against cancer and other diseases of aging beyond the age where most individuals would already be dead by other causes. And of course, I'm talking historically, I'm not talking in modern times. But for most of human history, survival past 50 years of age was rare. Less than 5% actually survived past 50. And so investing in that soma, if you think about the soma as being a vehicle for the germline, would have been largely a wasted investment. And that same energetic investment would have been better spent on somatic robustness that would, for example, help promote reproductive success. And I could give a whole lecture on just life history theory and how it influences disease risk, but we won't today. And so this graph is showing the percent of individuals alive for most of human history, and it's contrasting with modern incidences of cancer and heart disease and so forth. And so essentially, we think of these common curves for cancer, essentially the work of natural selection over millions of years, of basically sculpting a soma that limits the occurrence of cancers and other diseases of aging until this point where where those diseases were not having much of an effect on our fitness being our ability to pass our genes on to subsequent generations. And I think for this reason, life is not linear. And if you look at this curve of human survival, what's really interesting is, and this is in a modern industrialized country, is that from about the age of 5 to almost the age of 40, your chance of dying every year is about the same. It's less than 1 in 1,000. And most of that death will be due to, intrinsic reasons, right? Sort of intrinsic failure type reasons. Actually, when you're young, a lot of that death can be due to accidents and other things like that. But most human deaths in a modern industrialized nations are due to essentially what you can think of as intrinsic failures of the system, whether it's cancer, heart disease, neurodegeneration, and so forth. But there's a relatively flat period there, which actually is pretty amazing, because when you're 35, you're 30 years closer to the grave than when you were 5 years old. And yet your chance of dying in that year is no greater than it was when you were 5. But starting in your late 30s, with every subsequent year, your chance of dying is greater than your previous year. And in fact, it's an exponential curve. We don't just go downhill, we pick up speed. So, and you can even see it when you meet people. This is why 35-year-olds can play high school students, but a 45-year-old is never going to be able to play a high school student, right? I'm talking acting now, but anyway, you see it even in your friends. If you meet a friend again that you knew when you were 20, you meet them when you're 30, they don't look that different. But you meet someone when, you see someone when they're 50 and you don't see them again until they're 60, you can notice a difference and so forth. And it gets even more extreme as we go along. And of course, this is the waning of this pressure of natural selection to maintain the soma that really wanes when we get into the second half of our potential lifespans. And this is despite the fact, which I find really interesting, that most measures of damage, like, for example, DNA damage or mutations, for example, are relatively linear from maturity on. And what I think this tells us is that there's got to be some measure of resilience. And I think of the 3 R's of resiliency, in terms of sort of redundancy, and you get replacement, and you get repair. And this is what's maintaining our soma. And of course, it's an energetic investment, but it helps maintain our soma such that during that flat period, we maintain a high ability to pass our genes on to future generations. So then how are tissues maintained? And I won't go into all the details here, but you have everything from cell intrinsic mechanisms like repair to elimination of damaged cells through apoptosis, senescence. We have immune surveillance mechanisms, not just adaptive immunity, but for example, macrophages and so forth that can recognize a damaged cell and help eliminate it. Have peer pressure, where even a cell that might have a malignant phenotype, if it were placed on its own, in the presence of normal cells around it, will basically behave normally and contribute to the normal function of that tissue. And then we have one of my favorite processes, which is cell competition, whereby neighboring cells can actually force out a cell that is either poorly behaving, meaning it doesn't function as well for the tissue, or even one that is potentially malignant. So it's sort of a quorum, no, more of I'm forgetting the word, but anyway, so it's a quality control mechanism. And why does this matter? Well, a youthful soma essentially promotes our fitness. It promotes our robustness, maintains good tissue function, immune defense, disease avoidance, all those things that are happening during that first part of our life where we're relatively resilient in the face of diseases and other threats. And of course, it's also an anti-cancer defense. And that's what I'll spend a lot of the time talking today. But I want you to keep in mind that these mechanisms aren't just about cancer. They're about maintaining our soma and our robustness through these youthful periods, let's say up to about 40. So the model that we've been building that I won't dwell too much on is that cancer incidence has really been shaped by this sort of aging-dependent changes in drift, stabilizing selection, and positive selection, such that when we're young, that youthful tissue organization is very good at suppressing the expansion of potentially dangerous clones. And I'm using the word dangerous and not malignant on purpose because I don't think it's just about suppressing malignant clones, but also clones that could reduce the functionality of a tissue as well. And that suppression it holds through youth because that's where the investment was made to maximize our reproductive success. And in older ages, that can switch, and some of these same mutations can become positively selected in old age. And that's not because there's some mechanism to get rid of us. I don't believe in programmed aging, and I can go into that if anyone's wondering later why that wouldn't be the case. But it's more the waning of mechanisms to maintain us. The lack of selective pressure over millions of years to maintain that soma for those older ages because it was very unlikely that you were going to be contributing to the next generation. So we'll talk a little bit about clonal metapoiesis. So clonal metapoiesis is known to increase exponentially in old age, just like acute myeloid leukemia and myelodysplastic syndromes increase exponentially in old age. And what's interesting is when they've looked for clonal metapoiesis, when they first characterized it, you really only see this late life portion. In fact, most clonal metapoiesis would be what could be picked up by whole genome or whole exome sequencing. But the more sensitive the method, the earlier they can pick up these expansions, such that, if you use really sensitive methods, basically everyone has clonal metapoiesis when you're young. And it's really a question of whether these clones expand to the point that they could be detected by sort of cruder methods. And a lot of mutations have been characterized in clonal metapoiesis. And these different mutations can have different risk associated with them. First off, they're associated with different factors. They're pretty much all associated with aging, right? That's highly significant. But some can be associated with smoking or other types of exposures. But what's interesting is they have different risk of malignancies. And so, for example, if you look at DNMT3A, the risk of malignancies for most of these mutations is relatively modest. And then for TET2, it's a little bit higher for risk for malignancy, but still relatively low in the long run. But then if you look at splicing factor mutations, they're highly associated with the risk of malignancy. And so different mutations seem to come along with different risk. And this is going to become relevant later on. And so CHIP is also not just associated with leukemia risk. In fact, if you've got CHIP, that's probably number 3 or 4 in your list of things to worry about. It's much more likely that you're going to die of something else associated with that CHIP mutation, like coronary cardiovascular disease, or it's associated with gout, with liver dysfunctions, and so forth. And so it's still, I think, debatable to what extent is clonal metapoiesis associated with these different disorders, because it's a common manifestation of the aging soma, and to what extent those clones are actually contributing to diseases. And I think the evidence is pointing to both, basically, that aging promotes clonal metapoiesis, and clonal metapoiesis itself promotes some of these aging disease risk. And so this just shows you some of these associated disease risk. And what I'd like to point out is if you think about the TET2 mutation, it increases your chance of AML about 8-fold and your chance of cardiovascular disease about, a couple-fold, 2-fold. But actually that increased risk of cardiovascular disease is much more impactful because your overall per year risk of cardiovascular disease is about 250 times your overall per year risk of AML. So doubling something common is much worse than 8 timesing something that's very uncommon. So clonal hematopoiesis, as I mentioned, is very much associated with aging, and mice have been used to study this. And I think we can look at a number of factors. It's not just the passage of time, but you're getting a lot of changes throughout life in, the microbial content of the mouse. You're getting changes due to infections and so forth, particularly in the real world, less so in our mouse colonies. And that these could be contributing to changes in clonal dynamics. And these changes could be due to both cell intrinsic accumulation of changes within the hematopoietic stem cell pool, instituted by the external environment, I should mention, as well as changes to the niche, to the other cells, and to the overall environment that these cells experience. And so we wanted to model this in mice, and so a very talented postdoc in my lab, Marco De Domenici, set out to do this. And so he developed a very powerful protocol that is strikingly efficient. And basically what he does is he takes stem and progenitor cells from mice, and he uses a lentivirus to introduce in the guide RNA of interest. And he could do that with close to 100% efficiency. And then he electroporates in the Cas9 messenger RNA. And the reason this is important is because the Cas9 will be a sort of a hit and run type mechanism, and it doesn't hang around causing additional breaks over time. And then he grows these out a bit, and then he transplants them back into mice, and allows them to manipulate both the age of the donor cells and the age of the recipient mice. And this just shows that the CRISPR is very efficient. He gets, about 2/3 of the cells will have a frame shift. And the others probably have a mutation. It just doesn't actually shift the frame, but it still may disrupt the protein. And when he did this for DNMT3A, he got the surprising result that basically there is no aging dependence for DNMT3A mutations. We get just as much very low level expansion in the young as we do in the old background. And this is sort of quantified over here. And in some ways, this fits with the epidemiological data, which shows that DNMT3A mutations seem to just sort of arise slowly throughout life. And they don't show that striking age dependence that you get, particularly with the TET2 mutations and the splicing factor mutations. Splicing factor mutations in particular are pretty much not detected in young people and only seen in old age. But instead, when he then looked at TET2 using the same protocol, he gets very strong aging dependence, where the young is shown in light blue and the old is shown in dark blue. And this is over time in the peripheral blood. But what's interesting is if he looks in the stem cell compartment, he actually sees that the TET2 mutation is pretty much neutral for young, meaning right at 1, because this is the ratio over a control event. I should mention that we're always comparing to a mutation in the ROSA 26 locus as a control, which is basically still controls for the CRISPR event and everything else, but shouldn't disrupt a gene that should have any relevance to leukemia. But in the stem cells, we could still see strong selection in the aged, but essentially no selection in the young. And, before we had published a while ago, when we were looking at aging-dependent selection for RAS mutations and BCR-ABL mutations within hematopoietic progenitor cells in mice, we had found that that selection was purely due to the microenvironment. In other words, we could put the mutation into young stem cells, and it was the age of the recipient mouse that determined whether or not it expanded. So we thought the same was going to be true here, and it was absolutely the opposite. So in this case, when we put young cells into young mice, we don't see much expansion, or young cells into old mice, we also don't see much expansion. It's the age of the donor cell that determines this selection. And this is a real shocker for us because, as Jaime mentioned, I've spent my whole career saying it's all about the microenvironment. But I still think it's about the microenvironment. I think it's about the microenvironment that those cells sat in for 2 years in this mouse. I should mention all of our old mice are about 2 years old. I should have mentioned that earlier on. that has basically permanently rewired these cells. They're hardwired to not function that well. And in some ways that should have been obvious, because there's decades of research from like the Weissman lab and other labs showing that if you take old stem cells and you put them into a young mouse, they still function poorly. So you can't rejuvenate the stem cell by putting it into a young environment. And we see this expansion with TET2 mutation even by middle age. But still, it's only in the oldest mice where we get that expansion within the stem cell pool. So there seems to be differences in sort of the shorter term progenitors where we can get expansion even in middle age, and the longer term ones where that expansion seems to be mostly restricted. That selection appears mostly restricted to older ages. So we wanted to actually do like a fitness test. And we wanted to ask, is the relative expansion of these cells due to the fact that they're so awesome, or is it due to the fact that they're surrounded by sort of mediocre competitors? And I think it's kind of a good life lesson. If you want to excel in life, you have two choices. You could be awesome, or you could just surround yourself by mediocrity and you're going to look great. I don't really mean that, by the way. So, when we do this test, we find, for example, as we expected, the TET2 knockout cells have a clear fitness advantage over old wild type cells. So we've set up different competitions within these mice. And then if we take the old TET2 knockout cells, they have a higher fitness than young wild type cells, but not nearly as much. So just having young wild type competitor cells almost suppresses that clonal hematopoiesis. But the old TET2 knockout cells have actually a lower fitness than young TET2 knockout cells. So it's not like that combination of TET2 knockout plus old hematopoiesis has created a supercell. No, it's just that they're competing with crappy old cells. And finally, the old wild type cells have a much lower fitness than young wild type cells. So really, the reason we see this competitive expansion is basically because of the relative fitness differential. And the relative fitness differential between an old TET2 knockout cell and an old wild type cell is much greater than what you would get within the young. And so we also wanted to ask, well, we know that when we get old and when mice get old, their hematopoiesis becomes much more myeloid biased. Again, work from Weissman and others over the decades. And that's in part because you have actually a change in the stem cell composition. We have more myeloid biased stem cells. So Marco sorted out myeloid biased stem cells and lymphoid biased stem cells separately and repeated the experiments I've shown you, looking at the adaptiveness of TET2 mutations in these different backgrounds. And he also sorted out like some of the short-term stem cells and myeloid, the multipotent progenitors, which are more committed, to ask, could we see adaptation within those compartments as well? And what was pretty clear is that we see adaptation in both stem cell compartments. It's quite equal. So it doesn't appear to be because of the myeloid bias that we're getting this increased adaptation. And I'll note that we still saw myeloid biased hematopoiesis when we put in myeloid biased stem cells. So he sorted the cells correctly. But what was a real shocker from this experiment is that it's been known for a long time that if you sort out multipotent progenitors and you put them into recipient mice, those progenitors basically can contribute to hematopoiesis for a couple months, but then that contribution wanes away. And you could see that over here in the wild type, in the sense that over the time course of this experiment, basically contributions to hematopoiesis basically go to zero. But in the ones where we put in TET2, we maintain hematopoiesis quite dramatically. And so it appears that the TET2 mutation is conferring some level of self-renewal to these committed progenitors, which may be how it's even providing an advantage to these progenitors in, for example, like middle age or even to some extent young mice. And by the way, you can still see with the stem cells transplanted that you get this big differential between the young and the old in terms of their adaptiveness. And you can see this within sorted stem cell populations as well. So then the question is why? Why is TET2 mutation selectively adaptive in this aged context? And of course, when you don't have a clue, you do omics. And so we did single cell RNA-seq of these cells. And we also did some of the more mature cells, but I won't get into that for this talk. But we get a nice pattern of different stem cells and progenitor cells that make up the hematopoietic system. And we've got both the TET2 mutant and the wild type within that. And we've collaborated with Sheng Li's group and Lamis in her lab for the analyses of these data. And so what was really striking, and this is really shown here, where this is and I'll have to put my glasses on to see this a little better. That's one of the factors of aging, is that if you look at the genes that are upregulated with aging, they're shown in the red, and they're uniformly suppressed by TET2 mutation. So genes that would normally be overabundant in age relative to young are pretty much almost entirely suppressed. So TET2 is doing an amazing job at rejuvenating hematopoiesis. And if you look at all other genes, you can see that they're hardly changed by TET2 mutation. So it seems to be pretty selective to the stuff that is upregulated in aging. And you can see this with these ribosomal protein genes, and we see the same thing with a lot of mitochondrial respiration genes, is that you get this increase in aged that is largely suppressed by the TET2 knockout. And when we look at the pathways that this is regulating, we get a lot of interesting pathways that you might normally think, well, shouldn't this be associated with increased fitness, like more ribosomal genes? Except it's been shown that ribosomal proteins can promote, excesses in ribosomal proteins can lead to sort of a stress that promotes p53-dependent cell cycle arrest or even cell death. And in fact, actually, some of the top genes that we observed are in the RUNX pathway, which is known to regulate a lot of these ribosomal transcripts, and in the p53 response pathway. And so essentially the idea being that TET2 with RUNX is promoting an excess of some of these ribosomal proteins that no longer can be properly incorporated into ribosomes. And these have been shown by other groups to inhibit MDM2 inhibition of p53, thus activating p53. And so then we ask, well, if that's the case, should losing RUNX1 or losing p53 themselves be adaptive in the old microenvironment? And that is true for RUNX1 loss, where you can see that knocking out RUNX1 in young is not adaptive, but in the dark green, knocking out RUNX1 in the old is more adaptive. But what was really cool is here again is the selection for a TET2 mutation within the old background. But if we combine in stem cells where we've knocked out RUNX, we can largely suppress that expansion. So in other words, we can, by boosting the competitiveness of the other cells, we can suppress TET2 mutant expansions. And we basically do the same thing with p53 loss, which is shown over on the right. And also I'll note that that is observed in the HSC compartment and when we actually sacrifice these mice. So again, it's a relative competition, but it seems that the reason that TET2 mutation is adaptive in these cells is because it's suppressing this RUNX1 p53 type of response through ribosomal gene expression. So just to summarize what I've shown you so far is the TET2 knockout cells expand at a faster rate in aged mice compared to young mice. The rate of this expansion is dependent on the age of the donor and not the recipient. So it's cell HSC intrinsic at least. And the acceleration of TET2 knockout expansion is noticeable even by middle age in these mice at about 7 months of both male and female mice. The TET2 knockout seems to confer some increase in self-renewal property to multipotent progenitors. And the expansion of the TET2 knockout is due to this reduced fitness of the old cells. And what I showed you is that it's this fitness differential that appears to be key. So if we think about this, well, what is life? What is happening throughout life? And certainly this is different in, mice in our clean environment, in the clean cages that we keep them in versus real life. But I think particularly in real life, it's not just the normal aging process, it's all of our exposures. And one of the things we've become very interested in is this potential that repeated infections throughout life, by sort of altering the inflammatory milieu, could actually be contributing to this ratcheting effect of our stem cells through life. And that we've evolved the resiliency of our stem cells to tolerate this to at least the point that we maintain good, healthy stem cells, again, through our reproductive years. And so that's something that I think we're very interested in pursuing in the future. So I'm going to briefly turn to a third mutation, the splicing factor mutations, and just going to present a little bit of data on this because I want to get to the lung. And so, as I mentioned before, U2AF mutations are really potent and they're highly associated with leukemia incidence. They're also not the most common ones, probably not accidentally, but when you have these mutations in clonal hematopoiesis, it's a very strong risk factor for leukemia progression. And it's known that these disrupt the normal splicing machinery in sort of a dominant fashion. So these are always heterozygous mutations. In fact, the same mutation in a homozygous form is lethal to the cell. So it's a alteration of splicing. It's not simply a disruption of splicing. I mean, it's not an elimination of splicing, obviously. And with our collaborators, we've looked at some of the genes that are changed in terms of splicing. Exon inclusion and exclusions are changed, but I won't get into that. But I think it's important to note that it's a high-risk gene, which contrasts with DNMT3A, which is a very low-risk gene, and TET2, which is sort of a more moderate-risk gene. And so what was interesting is when Bridget did some of these very similar experiments, now she's using actually genetically modified mice, and she can induce this mutation in a small fraction of the cells. She found that we get very strong selection against the mutation in the young background. And note that this is a log 10 scale. So this is really, they're just being driven to extinction within these mice. And this fits with the human data where it's really even hard to find these mutations in young people. We've even used duplex sequencing and we can't see them. But what's really interesting is when we do this sort of young into young versus old into young, et cetera, it's only in the old into old background where that gets reversed. And we now see positive selection for these very same mutations. Same mutations, but the age of both the donor cells and the recipient cells seems to be important. And you'll note that when we do the other crosses, we get kind of intermediate effects. So if we do old into young, so the old donor cells, we can see much less negative selection, but still a little bit of negative selection. And then if we do young into old, we basically no longer see significant negative selection. So there seems to be both a cell intrinsic and an extrinsic factor. And I think this is interesting because as I've been saying, depending on the model we use, we either get a very strong component that's cell intrinsic, extrinsic, and now both. And it sort of suggests that natural selection's goal doesn't really have a goal, but what is the sort of the currency of natural selection is reproductive success. But the way that is achieved is by not having death during youth. And I think natural selection uses many different mechanisms to achieve that. And so if we go back to that mathematical modeling that Andre Rosak did years ago, and we sort of now think about these different, curves, we can now think of like the really strongly selected against curve as being the splicing factor mutation curve. And the ones in between could be more like the TET2 mutation curve. And then maybe the DNMT3A mutation curve is a sort of a light blue curve where not much selection against it in the youth because its risks are minimal. So essentially, natural selection has pushed down on these different mutations to the extent that benefits survival. And Andre had modeled this or put a model into the end of the paper at the very end where he talked about this in terms of the number of mutations, where if a malignancy required only one mutation, there'd be really strong selection against that expansion in youth. But if a malignancy required multiple mutations, you could have weaker selection against it. But I think we could envision the same thing being true even for just individual mutations. That how dangerous a mutation is going to determine how much natural selection pushes down on it in youth. And again, we can sort of affix these mutations very lightly to these curves. So with that, any questions so far? Everyone needs a break. Yeah, Aaron.
Could these age-associated mutations just be the presence and absence of, say, P53 or other protective mechanisms? Like if you have a KRAS mutation in you, it's suppressed by the presence of an active P53. Could those mechanisms be sort of
Excuse me, could you please repeat the question?
Oh,
Sorry, we need microphones for questions in the room.
right. I'm sorry, I didn't realize. Yeah, that's true in the room. Yeah. So the question from Aaron is whether these could reflect changes in sort of tumor suppressive capabilities, like such as through p53. I certainly think it could reflect differences in general tumor suppressive mechanisms. I'm not sure about p53 in particular. There was some report ages ago that there was, like that there was more p53 activity in or reduced p53 activity in an old context. And we actually see the opposite. We see more p53 activity, but we think that's going to lead to stronger selection for loss of the p53 pathway. It's going to sort of be the opposite. But I think in terms of, like, for example, mechanisms to get rid of cells that might have these mutations, I think those definitely go down in old age. Just the quality control mechanisms go down in old age. So that could, I think, definitely play a part in it. And of course, I think that because of the change in the tissue microenvironment, these mutations can simply be more adaptive. So this is no different than animal evolution, where, over long periods of time, animals, plants, et cetera, don't tend to change as long as their environment doesn't change, because they've reached a point where they're at a fitness maximum. And so change tends to be bad. It tends to reduce fitness. But if you get a change to the environment, like when the meteor hit off the Yucatan 65 million years ago, and thanks to that meteor, we're having this conversation, that you now created new niches and new environments that could stimulate evolution of new types adaptive to the new environment. And I think that's kind of what's happening within our bodies. Now, that happens naturally and more gradually over aging, but I think that can also happen in terms of, why is chemotherapy associated with secondary malignancies and so forth, right? It's not just the mutagenic properties of chemotherapy, it's the fact that it's dramatically changing tissue environments.
Super interesting. So, with regard to TET2 loss, I would think that in people, you first have mutation of one allele, later loss of the other one. And the way you did the experiments, you're getting both at once. And I'm wondering, is there haploinsufficient effect?
We have an experiment ongoing where we had to use a genetically modified mouse to be able to do that experiment. That's a great point, because CRISPR, you can't stop it. Right. And most of those mutations are probably going to be biallelic. And yeah, in humans, they're probably mostly monoallelic. What has been shown is that the actual effect on the cell seems to be pretty similar for the monoallelic and biallelic, which I've never quite understood. Yeah. But it does appear to be the case that it's haploinsufficient to the point of being almost a null when you lose one copy.
Some kind of tipping point.
So we think that the model is still legitimate, but it is imperfect in that respect. So we actually have an experiment ongoing. We had to age those damn gems. That was the problem. So, but yeah, I agree completely.
So, one last question. One thing that we know seems to affect aging, even, possibly in primates like us, is caloric restriction.
Uh-huh.
So have you or others shown that caloric restriction can change the environment in such a way that it changes the selective potential of some of these mutant clones?
We have not tested caloric restriction. We did try rapamycin.
Yeah.
And we showed the rapamycin worked, and it wasn't sufficient to reduce the clonal hematopoiesis. But, and as someone who likes to eat, but also likes to exercise, we went with exercise. And so I actually pulled it from the talk because I thought the talk was going to be too long. Uh-huh. But we actually have shown, we give the mice running wheels. First off, they run 10 to 15 kilometers each per night. It's amazing. The lights go off and they just start running, and they don't stop till the lights go on. And we have reduced significantly clonal hematopoiesis with exercise.
Wow.
So, who really wants to calorically restrict? Jaime.
So real quick, there's been a lot of attention to this change in the relative proportion of myeloid and lymphoid cells associated with a lot of diseases, including many cancers.
Yeah.
And clearly it's associated with aging. But I wonder whether you think that there's actually a systemic sensor. So that's associated with the lifespan of these cells. So lymphoid cells generally tend to be really long-lived, right? Whereas myeloid cells generally are short-lived. So I wonder if the bias in the hematopoietic cells is associated with some sort of sensor mechanism that says there's enough lymphocytes and lymphoid tissue,
Yeah,
but our neutrophils keep turning over.
yeah, no,
I don't know if that makes sense in terms of the way that we age, not necessarily being confined to just a single organ or tissue, but really being the whole organism.
yeah, no, I think there's definitely some, I mean, there's evidence that the myeloid bias in hematopoiesis, first off, it starts at an age where you would still be subjective to natural selection, right? It's in, early, you're probably, I don't know in humans, but in mice, by 5, 6 months, the first maybe 7, 8 months, they start seeing this myeloid bias. So it's probably that like you said, you need to generate your army of lymphocytes. But once you've generated that repertoire, there's still some generation going on, but you don't need as much of it. You've already had all the recruitment. But myelopoiesis is your first line of defense. And, that you need to continually keep up. And that may become more important as we age. So that's more of a program and less of a sort of you're beyond the realm of natural selection type change. But I do think it influences disease predisposition in old age. And for the reasons that you just talked about, the sort of increased inflammation and so forth. But these are all programs that were very useful when we were young and probably even remain useful when you're old. It's one of the reasons I think we have to be very careful with like anti-inflammatories and stuff like that. They did a study with blocking IL-1, reduced lung cancer risk 3-fold. You think this is great, or this is mostly smokers. And it turned out there was no increase in overall survival because people died of sepsis. So, there are reasons we have all these programs. Yeah, right here.
Just kind of follow up with your transplant experiment. Is that like a primerase transplant? Have you done serial transplant to actually see whether the mutant cells actually have the ability to like self-renewal after serial transplantation?
we haven't done that with TET2. We did do it DNMT3A, and they did persist, but we still didn't see an aging dependence. We thought we might see it there, but so we haven't done it with TET2 yet, though. We need to.
And then,
yeah.
sorry. So just to kind of clarify, so what you're saying is that in the TET2 model and the UTF1 mutant models with the aging kind of cells, are you saying that there's some sort of intrinsic sort of cellular changes within those aged cells that's
exactly
giving them that like fitness, right?
Yeah, exactly. So TET2 mutation is adaptive in the old cells, even when taken out of the old mouse,
right? Even
when we put them back into a young mouse. So there's been some permanent ratcheting of the fitness of those cells. They're less functional because of their intrinsic abilities. And if you don't mind holding that, because I'm going to try to get through this last bit, and then we can have more questions at the end. And so just to kind of get into this last bit, there's been a lot of work recently showing how our tissues become dominated by these these clones driven by cancer-associated mutations as we age. And this is just a few of those papers. And so Ed in my lab actually did this was actually during the pandemic when, he couldn't come into the lab, but he's got computational chops. So he did sort of a computational meta-analysis of all these different studies and basically used the same methods to categorize all these different mutations and how they were being selected. And what he finds is by the time you're in your 60s, you've got roughly 100 billion cells in your body driven by known cancer-associated mutations. And this is just in the tissues that have been analyzed to date. And this was just with the methods that have been used to date, which are relatively insensitive methods. So this is probably a gross underestimate of that number. And that's out of 3 to 5 trillion, nucleated cells within your body. So it's a pretty big chunk of your cells have known cancer-associated mutations. And yet only, 40% of us will develop a cancer, and that's going to rise from one of those 100 billion or so mutations. That raises the question, what are all those other cells doing in our bodies? And what are the consequences of all these clonal expansions within our bodies? And this is sort of what it looks like in the esophagus. Basically, your esophagus becomes more occupied by these clonal expansions than not. So we've been asking this question for the lung. And these are some of the collaborators. It's a long-term collaboration now with Charlie Swanton's group and Miriam Hanjani, as well as more recently with Nuria's group from Spain. And so we've used different cohorts. So this is all going to be on normal lungs. And I should put normal in air quotes because You can't really get normal lungs. So for example, from our Colorado cohort, these are from people that came in for bronchoscopies. And they came in for the bronchoscopy because they had something detected by CT that was suspicious. But in over half the cases, turns out there was nothing. And we actually have gotten a biopsy from a different lobe, so a completely different lung. So it's quite far from any potential cancer. And I can tell you that whether or not they had cancer had no influence on the spectrum that we observed over in the other lung. And we also get them from the Tracer X cohort and the Peace cohort in England. Tracer X are biopsies that are close to, but separated and morphologically normal from a cancer. And the Peace study are autopsies of people that died of cancer, but we purposely picked mostly samples that came from people that did not have any lung involvement. So again, not totally normal, but relatively normal. And then we use duplex sequencing. And in short, duplex sequencing is highly error-corrected sequencing. And in part, this is not just because we use unique molecular identifiers, but because you separately capture and sequence the 2 strands of DNA. And so you only count a variant if it was present on both strands of the DNA. And so this reduces errors to below 10 to the -7th. And we're sequencing to get about almost 20,000 duplex depth. So we can detect a mutation well below 10 to the -4th, or even sometimes down to almost 10 to the -5th. And when we do this on these different samples, and this is just showing you the Peace cohort, you can see that across the 30 genes that we cover in our panel, that we get dozens up to hundreds of mutations per sample. And these include, single nucleotide variants and then small changes, indels and other small changes. We won't get the big changes because this is short read sequencing. And if we just look across our 3 cohorts, you can see we got a couple hundred samples in all, and we get differences in the number of mutations detected, but sometimes that's due to the depth of coverage, so don't take that too seriously. But you can see that if we look at cosmic mutations, which are mutations that have been shown to be involved in cancer, so it's a catalog of mutations, you can see the top genes that we see mutated are kind of the ones you'd expect for cancers, like p53, PIK3CA, and so forth, KRAS. But what I show you is that there's some really interesting things that pop out from this. So for example, if we look at KRAS, we get lots of different activating mutations in KRAS, and they're shown over here. And you can see that the variant allele frequency is higher in the smokers than the never smokers. So it's not just whether you get the mutation, which is probably increased by smoking, but how big those clones get. So there's both a component of mutation occurrence and selection for those mutations that's influenced by smoking. And we could see the same thing for EGFR. But one thing to note is that in lung cancers, almost all lung cancers are driven by the top 4 or 5 codon 12 mutations, and they rarely involve codon 61. And you see those codon 61 mutations being expanded in the normal lung, but with very little chance of ever ending up in a cancer. And so we sort of analyze this further. And so this is just showing these top, in the blue is basically those top mutations that are observed in lung cancer. You can see this in the Tracer X tumor studies that 80 or so percentage of the cancers have one of these top hotspot mutations in KRAS. But in our normal tissue, much more of these still cosmic designated mutations shown in gray are not the hotspot mutations. So these other mutations that can be found in some other cancer somewhere else, they're just not, they don't tend to be in the lung cancers. But the other thing to note is that the never smokers are very good at avoiding the true hotspots. So they'll get clonal expansions driven by KRAS mutations, but they tend to be the ones that have very little chance of actually ending up in a lung cancer. They might be a codon 61 mutation, and those are associated, for example, with multiple myeloma, but multiple myeloma is not a risk for a lung epithelial cell that gets a KRAS mutation. And even on a per gene basis, we can see big differences. So on the right in red are the lung cancer frequency of these mutations. And over here on in the blue is the frequency in the normal tissue. And you can see that for a lot of these genes shown with the star, they're significantly more represented in normal tissue than they are in the cancers. And in fact, for many of them down here, and again, this is only mutations that are found in some cancer somewhere. That's the cosmic designated. You can see a lot of these mutations, like in ATM, for example, or in ERBB4, where they're virtually never found in lung cancer, but they're quite common in the normal tissue. So they're adaptive in the normal tissue, but not contributing to malignancy. And this just shows you for specific driver mutations. In PIK3CA, normally in the cancers, there's 3 hotspots that represent over 80% of the mutations in these cancers. But in the normal tissue, you get much more of a distribution of these mutations. It's really across the gene. And when we map these onto the structure, in green is the regulatory subunit, you can see that these red hotspot mutations map to that interphase, and that's been shown before. But what was interesting is a lot of these yellow, more minor mutations that we find in the normal tissue also map to the interphase. So what they may be doing is doing something similar to the hotspot mutations, but perhaps not to the same extreme, where it's enough to be adaptive in the normal tissue, but not so much that it could contribute to a malignancy. So only a subset of mutation-driven clonal expansions have a likelihood of contributing to a cancer. And a mutation can be adaptive in normal tissue, but not contribute to malignancy. And finally, if you don't smoke, you're actually very good at selecting to not get the real bad boys, the real hotspot mutations. And so to quickly look at selection, we want to ask, what are these mutations being selected for? We're going to use D and DS. This is simply looking at mutations that change the amino acids versus those that don't. And that ratio can be determined whether something is under, a gene is under positive selection, is neutrally being selected, or be it under negative selection. And we've done this in Foz and Charlie's group came up with an algorithm to look at this where he combines it with the variant allele frequency. And you can see when we do this that now you can see some of these tumor suppressor genes a little better. And I'll mention, because if you do normal, if you actually ask whether they're cosmic designated, most of these mutations are not, because there's 1,000 ways to kill a tumor suppressor, right? Frame shifts and so forth. And so this is allowing us to look at selection for tumor suppressors without having to worry about it being a recurrent mutation. And you can see NF1, P53, and Notch are some of the top ones that come up. And if we look at the other cohorts, we get some of those same genes. So this consistency across the cohorts. And we've collaborated with Nuria Lopez-Bigas's group and the student in the lab, Feriol, to basically categorize these mutations further. And they can look at, whether there are excess mutations per type of mutations per gene. They look at 3D clustering based on AlphaFold to ask whether there's a common location for these mutations in 3D space. And finally, they can look at the functional impact. In other words, how much are these expected to affect function of the gene? And when you do this, you can see, and I'm drawing these lines because I have trouble following them up and down. You can see that, some of these genes that, we know and love, like BRAF and KRAS, so very strong selection for missense mutations, but of course not for truncating mutations. But in contrast, tumor suppressors like CHEK2, P10, and PP3, so very strong selection for the truncating mutations, as you might expect for a tumor suppressor gene. And when we compare the smokers to the never smokers, we can see that smoking increases selection for disruptive mutations in BRAF, CHEK2, and so forth. And you can see this really strongly for KRAS. We get much stronger selection in the missense mutations for KRAS. Interestingly, for P10, we see, for the missense mutations, we see smoking-dependent selection. I should mention smokers are the open bars, never smokers are the hashed bars, and if it's filled in, it's significant. But interestingly, we do see strong selection for P10 mutations and truncated mutations, even in never smokers. And we've seen this for a few other genes where only some of the genes show the smoking dependence of the selection. Other genes seem to be selected for kind of independent of smoking status. Probably just aging. I should mention pretty much everyone we're looking at is older. And you might wonder, how prevalent are these mutant clones? And so this is where we can basically just add up all the expansions in every individual, because sometimes you get multiple expansions of the same gene in a single biopsy. And when we do this and we compare it to our control gene, TM2, which tends to have expansions that are below 10 to the -4th, we can see that a lot of these genes are showing selective expansion, but only some of them are showing that selection in a smoking-dependent fashion. And this just adds up all expansions, asking, how much of your lung is occupied by these expansions? And if you're a never smoker, the variance allele frequency is about 2%. And if you're an ever smoker, it's about 9%. And if we assume that most of these mutations are heterozygous, which they probably are, that means that about 4% of the lungs of a never smoker and about 18% of the lungs of a smoker are occupied by these clonal expansions. And again, that's probably an underestimate because we've only looked at our little panel of genes. But that could have effects on the functionality of the lung, not just cancer risk. And I'm going to skip the multi-region, but suffice it to say that if we looked at a bunch of pieces from the same individual, basically they show very similar patterns of mutations, such that on a PCA plot, all those pieces from the same person kind of cluster. So there seemed to be a flavor of selection in any given individual for directionality. And I'm not going to get into the functionality. So basically, what I've shown you is that smoking promotes selection for oncogenic variants in the lungs, but only a subset of these mutations will actually contribute to a lung cancer. And we observe pervasive positive selection on these mutations, sometimes in a smoking-dependent fashion. And that the adaptive landscape is specific to an individual's lungs. And then finally, eating pork rinds we've shown it completely prevents clonal expansions. I had to throw it an April Fool's joke. No one noticed it was April Fool. So just to kind of sum up the model here, that, what we're basically saying that when you're young, your healthy tissue is very good at maintaining the status quo. It impedes these clonal expansions. And because these clonal expansions can themselves be disruptive of tissue, it helps maintain the normalcy that further impedes clonal expansion. So it's a positive feedback loop that maintains us. But we get old, or we do things like we smoke and other stuff. And this compromised tissue can select for harmful clonal expansions, which then changes the tissue that can help promote further selection for harmful clonal expansions. And this is why, as I mentioned, we don't just go downhill, we pick up speed. My apologies for anyone in their late 30s. Enjoy the time you have left. And so, of course, lifestyle exposures and genetics play into this, and they can influence these outcomes. And what I think is important, this is relevant not just for cancers, but for other diseases as well. And really, I think for aging in general. So I kind of thank people along the way as I've shown the work, but great collaborators and, in England and USC and so forth. I should mention all the artwork was done by my son, who lives in Portland. And he also did the artwork for my book, when some of these pictures were stolen from my book, and of course, all the funders and so forth. And my lab is not this big. It's just that this was actually a reunion that brought people back as well. And finally, I'll mention that I am editor-in-chief of a journal. If you've got anything related to aging and cancer, I've got like waivers I can give out for page costs and stuff, but we're definitely looking for good contributions from the community. And finally, go Nuggets.
Okay, I'd like to start with a question from our online audience. Dr. Farouk asks, what about the natural selection process in childhood cancer?
Yeah, no, it's funny they brought up childhood cancers. I think that there's a few exceptions to this aging rule, right? The rule where you see most cancers in old age. And there's three things about those cancers that I think are of interest. One is they tend to be rare. Natural selection is very good at avoiding cancers till old age, but it's not perfect because there are always trade-offs. So one potential trade-off is, you can imagine that, death from infection for a child was a way bigger risk than leukemia for the vast majority of our evolutionary history. And so if having a more ramped up immune system came with somewhat of a cost of a 1 in 3,000 risk of getting a leukemia, that was a cost that was basically well worth it, as tragic as childhood cancers are. The other thing is they tend to be associated with modern conditions. And so natural selection hasn't had time to catch up. In fact, there's work from Mel Greaves that has shown that childhood leukemia seem to be associated with more sort of hygienic environments, kind of falls into the hygiene hypothesis. And that part of the reason you get this peak in ALLs, I think about 3 to 5 years, is that it correlates with the kids suddenly getting exposures that they weren't exposed to earlier. And so that sort of inflammatory insult that they get might be promoting for some of the mutations that might already be there. In fact, having an older sibling reduces your leukemia risk. Getting vaccinated reduces your leukemia risk, and even going to preschool reduces your leukemia risk. So those are things that sort of are modern conditions. And the final factor that I think can contribute to childhood or to cancers in younger ages is when they're due to infections. Because obviously infectious organisms have their own evolutionary agenda and they're constantly in an evolutionary battle with us. That's my long answer.
Any other questions in the room today? All right, let's say thank you very much for a great talk. Thank you.
Thanks.